Hello everyone, my name is Dr. Mohammed Al-Gnaya. I am assistant professor at King Saud University, College of Pharmacy, Department of Pharmacology and Toxicology. Today I would like to share with you my research which is titled Assessing the Role of Store-Operated Calcium Entry in the Interaction between Macrophages and Breast Cancer Cells and the Subsequent Cancer Progression. So the basic idea behind our research is studying the interaction between macrophages and breast cancer cells, and whether this interaction can be interrupted or suppressed by store-operated calcium entry, which is the pathway that we are interested in our lab. So the main objectives of our study is to determine the effect of inhibiting SOC on raw macrophages cultured in 41 breast cancer cells associated condition media, and the second aim is to see the reverse, which is basically determining the effect of inhibiting SOC pathway on breast cancer cells cultured in macrophages associated condition media. So the main idea is trying to understand the role of store-operated calcium entry in the interaction between macrophages and breast cancer cells. So just briefly, the experimental design, what we will do in this project, or what we did, is to culture 41 cells, which are the breast cancer cells, leave them for 48 hours, and then collect the media and centrifuge, and then treat raw macrophages with this condition media, with or without the BTB2 drug, which is the SOC inhibitor that we are working with. And then raw macrophages, after 24 hours of treating condition media and BTB2, we will measure COX-2, interleukin-1 beta, and vascular endothelial growth factor, and we also count cells to measure the proliferation of the immune cells. And again, we will collect the media, centrifuge, and then treat the condition media from this cell line, which are the immune cells, with or without BTB2, which is the SOC inhibitor, and then we will treat them on 41 breast cancer cells. And then again, the second aim, we will measure COX-2, interleukin-1 beta, and VEGF on 41 of breast cancer cells, and then we will measure the proliferation. So in total, we will measure the genes and proliferation in raw macrophages, and we will do the same with 41 cells or the breast cancer cells. So the results, the first thing we looked at is to measure the proliferation, VEGF, interleukin-1 beta, and COX-2 in raw macrophages treated with condition media from breast cancer cells. And we have two different percentages, 50 and 100 condition media, and we see an increase with 50% in all of these markers, VEGF, interleukin-1 beta, COX-2, and the proliferation of raw macrophages. We see similar thing with 100, but in some of these markers we see a reduction. So basically we concluded that the optimal condition media, or the percentage of condition media we're going to use, will be 50%. So now we did the same thing. We treated raw macrophages with condition media from breast cancer cells, and then we also added BTB2 as the SOC inhibitor to see if inhibiting SOC will prevent condition media induced effect on immune cells. And we see the same thing. We see condition media increasing proliferation, VEGF, interleukin-1 beta, and COX-2, and most importantly, we see that BTB2 inhibit the condition media induced effects on raw macrophages. We also see similar thing in breast cancer cells. We see that condition media between 41 and macrophages increase in the proliferation, VEGF, interleukin-1 beta, and COX-2, and BTB2 block all of these effects. So basically what we see in this study is an evidence that targeting SOC can be a valuable therapeutic approach in preventing the interaction between breast cancer cells and immune cells and the subsequent cancer progression. Thank you all for your attention.